So I'm actually a graduate student. So I have my master's with Paolo Grigolini in physics, and I'm working on a biology PhD with Dr. Ione Hunt-von Herbing, who is also in the audience this year. And so because I have a very interdisciplinary PhD ahead of me, I wanted to show you how diverse my thesis committee is. So I have a bunch of books ranging from all the different topics of all the different people on my committee that I thought would be fun to share. So I have to take a lot of classes in biology, and these are the ones that I chose. Okay, so I'm going to just start off with some of my motivations. So my PhD is going to be seeking new knowledge between fundamental physics and biology. And so I'm looking for the underlying biological mechanisms using several different organisms for biophoton emission. So there's lentil seeds, which is the main focus for now, because I'm replicating Maurizio Benfato's experiment to constrain some of the information and relate it more to biology. Later, it would be good to go towards a different type of model organism. So biophotons have been typically done more in the plant realm more often, but it hasn't yet been done with many animals as far as I'm aware. So I could either look at zebrafish embryos. I'm also thinking about maybe like a squid, so that there are just new sources of information about the biophoton emissions. So you might ask, what could we learn about developing organisms at both the macro and micro scales by using complexity measurements of biophoton emissions? And so here on the right, you can see the paper that Maurizio Benfato and his collaborators did in 2021. And overlaid on top of it is just an idea of what those lentils may have been doing at the time steps at the x-axis there. So looking at Benfato's work, we could have this conjecture that the seed germination timing lines up just as in the picture. And it would be interesting to see if the biophoton emission spectrum correlates to specific developmental stages. And then I'd like to find certain experiments that can determine the more accurate lentil seed germination timings. It turns out that's not a very studied thing. When you look at developmental biology for plants, they typically look at it forming on the plant, and they don't study when it's just a germinating seed. So as we move to animal organisms, it is actually very well characterized for every developmental stage. And so you can see that they have it down to the fraction of an hour for every cell division, all the way through the larval stage. They know very well those developmental stages. And because of that, if there is a correlation between biophoton emission and developmental processes, then it'd be better to use an organism where it's better defined than lentils. Here's another example of one that they have a lot of timings for, and so it's just a slower process for this squid. The zebrafish is a little bit quicker, but again, there's very specific timings of every developmental stage. Okay, so then what are biophotons and why do they matter? So it turns out everything alive is emitting biophotons. So you're doing it right now at this moment, and everything on this tree of life is also emitting biophotons while it's alive. So you might have remembered a quote from Star Wars where Yoda says, "Luminous beings are we." This is true. We are emitting an intrinsic light called biophotons. And so what they are is very weak photonic emissions. So this is just a few counts a second. It's in the visible spectrum. They are thought to be metabolically derived, and they are directly generated by living organisms between 200 nanometers and 800 nanometers. And what they are not is, these are not photons that come from bioluminescence, so they don't glow. They are not phosphorescent, so there's no absorption and re-emission as far as what's generating these. And they are also not thermal radiation, so this is not from heat. So we might ask, what can we learn about them using complexity? Because this is what my background is. And so when Biophoton's group did this paper, they actually looked at the complexity, so the statistics of this biophoton emission spectrum, and there was a distinct difference between, at some transition, a transition from crucial events, so a dynamic origin of complexity, to non-crucial events, which has a stationary process origin. And so my conjecture then is that at this transition point, there is some developmental marker that we can look at. And I just looked at the average time it takes to germinate a seed, lentil seed, and it turned out that that's about the right time. And so later on, I'll talk about that more. So this is significant because you have this renewal process, and then it transitions into a stationary process. What's special about this is that Paulo has been talking about crucial events for many years, and it's often confused with fractional Brownian motion, and this appears to be an experimental evidence that supports that crucial events are indeed a different type of complexity than one that is a stationary process. So renewal process, stationary process, they're different, but they're both complex. So he already was nice enough to go over, the math of this, so I'm gonna just skip through this. Here's some of the values that Maurizio found. I'll just skip through this. Just think of it as a measure of complexity. So I'm going to look at some other labs that study biophotons, just to give you a little background. So I mentioned that it's often studied in plants. And so there was a study done that showed infected plants where the emission would go higher when it was struggling, and at a certain point when it struggled too much, it would go down. And so you might think of it as, if you exercise, you're getting a good stress, and it's healthy for you, but it is difficult. But if you get a poison, that's bad. And so having this question of, is this stress after the fractional Brownian motion section is starting, or is it a more fundamental biological process in this case? We don't know. Oh, and the other one with the Kobayashi lab is just interesting because somebody was holding a cigarette and you could see the stress induced by the holding of that cigarette. So that's what the red area is. Okay, so we might talk about stressors, stress, and vitality. So you might say stress is bad, but again, you might have good stress. So we'll call those stressors in general. You could have where the biophoton emission is above a certain baseline due to a stressor that is maybe good or bad. If it's bad stress, it will dip below the baseline. Okay, so as I start my PhD thesis methodology, we'll look at some of the ideas I have to further Biophoton's work. So this was the experimental setup he had. So all I'm going to do is take what he had to start with and just go further, because those seed stages right there were never actually validated. And so we need to do that, because if my conjecture is correct, then I need evidence that it is. So all it was that I took the basic timings of seed germination of lentils and mapped those onto his biophoton emission. and saw that the radical emergence was the clear delineation point. I'll go into that more in a moment. So first, let's learn a little bit about seeds. Some of the anatomy that you should know is there's this thing called the radicle that will become the root, and this thing that becomes the plumule becomes the leaves, and then the cotyledon is like food storage, kind of like the yolk of an egg. You don't have to know too much about that, but at least know that the radicle becomes the root. And so during germination, it's this whole process that has multiple sections. So imbibition is the point where you wake up the seed with water, and then you have a lag phase where it doesn't appear like anything is going on, but it turns out there's a lot of internal processes going on during that time. And at the end of this second phase, you have the germination event where the radicle emerges from the seed. And it's at this point that the conjecture is that the complexity changes from that non-stationary process to the stationary process, both being complex, but different types of complexity. And then there's the phase 3, which is just growth. And so if the conjecture is correct, the fractional Brownian motion is occurring in the growth stage, and the crucial events come before that, between imbibition and the radical emergence. So as we look at what the biology of his experiment is, I just wanted to go over that I'm suggesting there are developmental markers that may be correlated to the biophoton emission spectrum. And so as you go through developmental time and intensity of emission, you can read that on the graph. You might be able to look at different effects of stressors like pH or moisture and soil characteristics. Those might change. So one of the questions is, does that fractional Brownian motion section have to do with a stressor, or is there a fundamental biological reason for that event? There's another problem with the data that we're not sure about. There's this area right after you close the box. It's just a few photons a second. So it's possible that there is some residual luminescence that we haven't been able to constrain there. I'm not entirely sure what to do with that yet. So my goal is to just focus on the developmental biology for now. But there may be experiments I could do to try and constrain what's causing that residual luminescence at the beginning, or maybe it is a feature of the spectrum of a lentil, I'm not sure. But the idea is that if I create a multi-box setup, I can start to look at some of these effects, because I want to sort of isolate the actual seeds, which was not done previously, and slice into them and see what developmental stage those seeds are at. So the statistics is just done using diffusion entropy analysis. And then I'm going to bin it with the developmental markers using this multi-box setup data results. And then hopefully that will allow me to determine if those are biological events that are correlated to developmental markers, or if it's some sort of stress effect that I need further experiments for. So the idea here is I have 2 photodetectors, whereas Maurizio only had 1. And so I'm just going to use that extra photodetector to create another box setup, just the same. Except this one, I'm going to remove the seeds just for simplicity's sake and slice into them. And so there may be some extra effects from removing them. And so then I have a third box to just check the growth. If the growth is consistent across all 3 boxes, then I may not need to do further experimentation. The other question with that, though, is does the spacing matter between the seeds? Because there could be communication, whether biochemical or otherwise, between the seeds. And then maybe the having less seeds over time might cause a problem. But I'll have the very first box to use with data in real time to kind of estimate the second curve. So at the end of everything, their growth will be measured, and the box where I'm removing it from would be the identical situation where I would expose the third box to light, just to make sure that the light exposure doesn't further complicate the situation with the experiment. And so I was just looking at some basic ideas of timing for this, because we do have this residual light curve, and I don't know if it's part of the experiment because it's a residual luminescence, or if it's something intrinsic to the biophotons. So to just get around it, if I measure every time I remove the seeds, if I measure every 2 to 5 hours to remove the seed, it should more or less equalize out that the residual light shouldn't be a problem. The other thing is checking for the crowding and spacing. So as I go down and remove seeds, I still want it to be a random sample, but I also want to make sure that the spacing doesn't go too wonky. So I have to figure out some kind of numerical calculation on just making sure that the spacing stays consistent. I may do further experiments with spacing just to make sure that that's not adding extra problems to my results. Okay, so just to summarize, biophotons have been considered as a sort of non-invasive imaging of stress for different situations, and no one really knows what their origin is, so finding this new way to characterize biological processes is pretty exciting. And then having this idea of the correlation of developmental markers might be really important for developmental biology, especially for organisms that are not yet described in terms of their developmental markers. And so I'm going to continue working with other organisms after I work with lentils. And I would also like to see if the lentils themselves were stressed rather than healthy, to further check if, this is a biological process, or if it is just from maybe those seeds were dying and that's what caused the FBM, because our previous evidence is that FBM occurs in stressful situations. But in the case of meditation, that's a good stress, so it's unclear if that's what it is, or if it's something to do with the growth process itself of the developing seeds. So the other thing would be to compare types of lentils that are organically grown versus commercially grown, because that could be part of the problem there. That could contribute to that biophoton emission change into FBM. And then after that, I have to start working, how do I modify the setup that Maurizio did to accommodate doing animal organisms, in this case, aquatic organisms. So just a few questions to think about for the future. Maybe biophoton emissions will someday be a way that you could use a quantitative measure of health or disease. There's a question about whether biophotons are actually just incidental products of metabolism. What actually are they? Do they indicate health or stress? And could they be co-opted through evolution for some sort of communicative purpose? Who knows? There's definitely a lot of different directions the literature has gone. So, but there is still a lot of room for new directions. Okay, that's it.
Thank you very much. One question? Okay. For you.
Okay, thank you. Very interesting. Finally, I understand something about biophoton. But the question for me is that there is a clear definition of biophotons. I mean, when you put your seed inside a black box, so I imagine that you define by photon what you find in that case, apart thermal radiation eventually, and discarding also bioluminescence if there is. When you measure biophoton from a hand or a plant, as you showed, in which way you can say, you can select a biophoton or filter biophoton from other radiation, and the intensity of biophotons that you define as biophoton compared with the intensity of a lot of other kind of radiation that came from the hand or from the plant. The relation between this intensity, which is, I mean, if you identify the biophoton, you have to say, okay, this is 1% of in the spectrum of I am observing and so on, something quantitative.
So yeah, the biophotons, you put a box and they radiate from the living thing. And so it's just a few counts a second. It seems, you know, we still have more testing to do about how size affects the emission rate. And I'm sure there's more in the literature about that. But if you eliminate the other possibilities of bioluminescence, phosphorescence, thermal radiation, then that's what's left. And so you can tell that, you know, we did dark counts and that sort of thing. So those are things to consider. But yeah, this is like 50 seeds and you've got like 50 photons at maximum. So this is, you know, not a large amount of counts. So you're just talking about a couple counts per second. And so if it is something that's a product of metabolism, then that's just the product of metabolism. Whatever the origin is, it's also known as ultra weak photonic emission because it is such a small count number. I don't, does that answer your question? Okay.